Ding, ding, ding. Let's get rolling. Not much time left. Gotta look at this one a little bit closely here. So we are never going to get here too far away from the central dogma. Here in this, we're going to talk about what might make this happen. This is transcription, right? This is transcriptional control because everything that happens in a cell, right? A trait, a phenotype, a kind of thing happening, whatever. Every kind of trait is due to proteins, almost totally, right? And this is the path that leads to proteins and traits, and we're going to look at, is this switch on or off, the transcriptional switch? Does it happen or not? That is the main regulatory mechanism by which all this DNA in a cell, which parts of it are being used at any one point in time to make RNA, to make proteins, to make a particular trait. So here we go. We talk about prokaryotes. They throw operons in here, much to your pleasure, right? So they're telling you an operon is a group of genes, right? It's transcribed all at once into one single mRNA. And this is an example of the kind that's called inducible, but they don't ask you for any more details about how it works. None of that gets in the blue boxes, right? Here we're describing a type of interaction that regulates gene expression. So I'm going to break it down for you a little bit, but since they don't in the blue boxes, they're not going to ask you these parts. An operon consists of those genes that were mentioned up here, a promoter region that's going to be mentioned later, and then an operator region. These are all parts of DNA, and all of these together make up an operon. So here's a picture of one, okay? Here's the promoter part, this section. Here's the operator part, that section. And these are all the genes, which are off the page here. These are all the genes that are either going to be transcribed or not, right? In this system called an operon. Another key aspect of this is a gene that makes RNA, that makes a protein that can repress or inhibit this, okay? This is a negative regulatory molecule, this repressor, because it inhibits, inhibits. How does it do that? By binding to DNA and blocking the thing that actually does the transcribing, this RNA polymerase, okay? Here they want you to note that there are sequences of DNA that don't code for the making of proteins, but they act to regulate that, turn it on and off, and how the location of those regulatory sequences relates to their function. So this promoter and operator, and the promoter is the only one that made the syllabus for you to know, but just so, these are in the beginning part. This set of genes is going to be transcribed in this direction. So having it in the location at the beginning means it's like a switch that can turn this on or off because it's at the start. So now, here, that repressor, right, which in the last picture was stuck right here, blocking this RNA polymerase, here it's now out of the way. Why is it out of the way? Because something has attached to it and changed its shape. That concept, that very key and basic concept, is easily worked into here. Why is this repressor not repressing? It's a different shape now. Why? It had something stuck to it. Okay, so what can this prokaryotic cell do now that it's making all this RNA that can make these enzymes? It can have the trait, it can do the phenotype of breaking down lactose in this case. And that's how the regulation of gene expression causes a difference in this cell from one point in time to another. Okay, so here we're at eukaryotes now, okay? So we're going to do the same thing for eukaryotes, and there are a lot of similarities. There's a little bit of a difference, but we're still talking about genes regulating the use, the regulation of the use of a gene, which is made of this chemical DNA, to be used to make RNA. This is what we're focusing on now, transcription, used again to make a protein to cause a phenotype. This gene expression is related to some kind of cell product it makes, some kind of function it does. That function is a phenotype or a trait. Central dogma never gets far away from this discussion. Okay, so here we have the idea right here that cell differentiation, right? Why are you a nerve cell, you a muscle cell, you an intestinal cell, even though you all trace back to this original cell called a zygote? It's because you use different genes. You had them regulated differently, okay? And this regulation is not just on or off, right? It's on or off for certain amounts of time. The level at which they are expressed means, are you on 60% of the time and off 40%? That means you're on more than off. And so it's not just simple black and white on or off. Okay, so right now in this eukaryotic cell, we are again at this part where this DNA is being transcribed into RNA, right? We talked about the signal that might start this whole thing. We're going to come back here later in the next video, and we're going to come back here in the next video. But right now, this is where we are. Again, transcriptional control in a eukaryotic cell now, not a prokaryotic cell. So here we have the same basic kind of idea. We have a promoter at the beginning of a stretch that's going to get transcribed. In a eukaryotic cell, that stretch has some parts that are going to be taken out. We're not talking about that gene regulation, but that's another place that this RNA could become different to make a different protein, different trait. Okay, so here we have the location of regulatory sequences that we did not see in a prokaryotic cell, and that is the ones called enhancers. They are somewhere upstream, somewhere different, right, than this unit of the gene and its promoter. And as you can see, a whole bunch of things have to attach here to make transcription happen. So those things are called transcription factors. They have to attach here at the promoter, and they have to attach here at this enhancer region so that it flips over and all of this connects to make this RNA polymerase able to transcribe. So real fast, I threw in here the fact that if these genes might be the kind that are regulating the cell cycle, some of them might make the cell cycle go. These are called oncogenes. Some of them might make the cell cycle stop. These would be called suppressor genes, tumor suppressor, because what is a group of cells dividing cancerous cells is called a tumor. So two nuns, a penguin, a giraffe, and a man with a parrot on his shoulder walked into a bar. The bartender said, "What is this, some kind of joke?" You see, and that actually is some kind of joke, because that, okay, I'm going to stop there.